Hello, my name's Ben McLaren. I work at Carnegie Mellon in the School of Computer Science in the Department of Biological Sciences. My colleagues and I are developing software designed to help students learn to think like a biologist. The curriculum is based on an idea we have come to call pathway algebra. The software that implements this curriculum uses a technology developed at Carnegie Mellon called Cognitive Tutoring. I want to give you a brief introduction to the tutor that we have designed, which we call the Cognitive Tutor for Analysis in Genetics, or CTAG for short. First off, let me say briefly that I can't emphasize enough that genetics is far more than simply a subdiscipline of biology. Genetics is the logical foundation of all of the biological sciences. It's also central to understanding health in the 21st century, as well as many other economically important fields. So the main purpose of this video is to give you a brief introduction to pathway algebra, but also some of the lessons that we have in the Cognitive Tutor for Analysis in Genetics. So to use CTAG, the first thing that you need to do is go to Carnegie Mellon's Open Learning Initiative website at oli.cmu.edu and sign up for an OLI account. It's very easy, it'll only take a few minutes, and register for the CTAG course. The course key, which you'll use for signing up, is CTAG S19 Guest, which is spelled exactly CTAG, C-T-A-G hyphen S19 hyphen GST. So when you sign up for the course and enter, the first thing you'll see is this syllabus for the course. As you use the tutor, you're never more than a few clicks away from this screen, so you can get your bearings and find out where you're trying to get to more easily. So the syllabus is broken down into units, the highest level of organization, and each unit is further broken down into modules. Let's start by clicking the first unit, appropriately named Unit 1. At this point, we can click on the unit bar to see the other units and move directly to them. You can also beneath the unit bar, we will see other modules within that unit that we can jump to. So let's go to the first page in the first unit. The first page of the introduction summarizes the 4 big ideas from the College Board's AP Biology curriculum. Even if you're not taking AP Biology, these concepts are tremendously important for anyone who wants to understand biology in the 21st century. They are organized from the most basic cellular processes to the most encompassing evolution. The 2 most relevant to us here are 2 and 3. Interactions and information flow. Number 2, interactions, says that the cellular processes that make up biological systems interact and that complex properties emerge from these interactions. The different states of the components of biological systems can be thought of as representing information. Number 3, information flow, says that these cellular interactions are connected and that these connections form directed networks that interact according to specific principles to retrieve, transmit, and respond to information essential to all living systems. The molecules in a living system and all of the different make up what is referred to as the interactome. A full understanding of the interactome is the holy grail of 21st century biology. The Human Genome Project was simply the first baby step towards this goal. As you move on and explore problem-solving activities in the rest of CTAG, I invite you to revisit these first few pages from time to time to remind yourselves of these big ideas. They will start making more and more sense as you learn the concepts and practice reasoning about biological systems within the pathway algebra framework. On the next page, page 3, which I get to by clicking this arrow up here, we see an explicit statement of the philosophy which guides the core curriculum of CTAG, which I call pathway algebra. Much of what students do in traditional biology classes, far too often still today, is declarative and memorization intensive. Students watch videos and learn to describe what happens in transcription or other important processes, but there is much more to being a biologist than this. Some of this is necessary and very useful, but an over-reliance on this approach in teaching and instruction means that opportunities for deeper learning by doing problem solving are passed over to leave room for simply more stuff. Pathway algebra, in contrast, is an abstract logical framework for representing biological systems. So we teach several topics using this framework, and within this framework you learn to formulate models or theories of biological processes. And these models support learning of two problem-solving activities central to being a biologist: prediction and explanation or experimentation. You can think of them as running a model forward or backward. Prediction involves determining what will happen or what you will observe if you know the underlying properties of the system. And explanation or experimentation is the process of inferring the underlying properties of a system given data and a theory of that system's behavior. That may sound complicated, but as students use CTAG, they come to see that as they come to see this idea as remarkably straightforward, or at least not as complicated as it might sound at this point. You will come to see pathway algebra as capturing the core thought processes of a biologist. CTAG attempts to put students in the driver's seat as much as possible, moving toward getting them to start building their own models of the world around them based on their observations of it. There are several statements or concepts that make up pathway algebra for biology. We call these postulates in much the same way that postulates form the foundation for geometry. They are based on key discoveries in the history of biology and are features of any area of study in biology. The most important organizing postulates here are the existence of two types of pathways. All systems in all biological systems can be thought of or represented as one of these two types of pathways or a combination thereof. From the simplest reactions all the way up to ecosystems and how organisms of different species interact with each other and the rest of the physical environment. The first type of pathway we call the assembly line pathway. These pathways take inputs and process them, building them up or breaking them down, or more commonly both in a coupled fashion, to get a required output. Example assembly line pathways include things from simple chemical reactions to much more complex processes like the Krebs cycle, glycolysis, fermentation, even transcription and translation. They all take inputs and produce the required outputs, mostly guided by enzymes or other gene products. The second type of pathway, we call it signaling pathways. These pathways are for communication and control. They involve gene products, usually proteins, that can be turned on or off in different conformations, for example, to trigger the next step in the signaling pathway. The different states of these gene products determine the output ultimately or the final signal in the pathway to turn on different genetic programs in the genome. So again, as I said, as the students use the tutor, these ideas which seem overwhelmingly abstract and complex will come to make far more sense, and students will learn to see this model of pathway algebra as a very, very general and transferable way of viewing living systems. So moving on to the next page of this introduction, we see a brief description of what the genetics cognitive tutor is. Then there's a pointer to a video that takes you through a few of the lessons and gives you an idea of how to solve problems in the cognitive tutor more effectively. Here's a brief outline of what cognitive tutor lessons, how they work. The basic idea is that there's a problem that is scaffolded or laid out for you with all of the steps in that problem, made explicit by text fields or pull-down menus, and then there is a help button that you can click on and get help and ask for more levels of help. There are several different levels of help at each step. You can go through this if you're interested. And the next page talks about what topics are covered in CTAG. There are currently 7 units that have been ported to OLI. And the most important ones, the most central to pathway analysis, are units 2 through 5, gene interaction pathways and epistasis and metabolic pathways, or assembly line pathways, I should say, gene regulation pathways, developmental pathways, and the development of the organism and development of an embryo, and cancer pathways. Each of these units has 2 tutor lessons. A forward reasoning, where you're predicting what's going to happen in a pathway if you know the underlying properties, and then a backward or experimental component where you're shown data and asked to run the pathway backward to determine what could have led to those observations. So each unit consists of declarative instruction describing the related genetics like a chapter in a textbook, and then after most of these units have a pretest or quizzes. Some of them have them in a couple places, but to help us understand better what students know at different points in using the tutor. Then a brief description of the tutoring activity, and then 2 or 3 CTAG tutor lessons, each with between 4 and 20 problems to solve, like the problems at the end of a chapter. So let's take a brief look at the declarative instruction for gene interaction to get an idea of what that looks like. So again, as I said, this is much like the chapter, the text in the chapter of a book that you read, which gives a lot of background information, all very important. And when I show you what the tutor lessons look like, it may not make a whole lot of sense, and that is, of course, because you have not read all of the declarative information all of the declarative instruction that goes into understanding the lesson. So one of the things that the gene interaction and epistasis lesson explains is how different pathways are affected by in crossing two parents with different genotypes that affect these pathways. One of the things you will note is that in the instruction there is often in several of these lessons some questions that are asked as you're reading through the material, just to encourage students to read more deeply what they're learning about, to better prepare them for the tutor lessons at the end of the chapter. This shows an example of what happens as students answer these questions about what they're reading about. If they are correct, the answer will turn green. If they are incorrect, the answer they select will turn red, with a description of why what they've selected is incorrect. After they read the declarative instruction, they will come to the cognitive tutor. So let's take a look at what some of these lessons look like. So here is an example of a cognitive tutor lesson. And this is gene interaction pathways, the forward modeling, the prediction. So the first thing the student does is they read the problem statement, which describes a pathway. The first thing the student does is they summarize the problem. This problem has been largely completed. Excuse me, just to show you what this activity looks like. So the first thing the student does is summarize the problem description. Then they determine the phenotypes for each of the true breeding genotypes described by this system. A true breeding genotype is one that is homozygous for both genes. Then after they do that, they examine crosses between two true breeding strains and look at to determine what the ratios would be in a case where you have what's called a dihybrid cross. Again, this will make more sense. Would make more sense if you read the declarative instruction. Several of the genotypes result in the same phenotype. We then collapse those and as the student is solving the problem, if they don't know what to do, they can ask for help by clicking the help button, and then get more help by clicking this, and eventually it will explain to them what they need to enter here, and this is the answer for that. And in one case you can get brown, and there are 3 of the 16 possible combinations are brown, and in this case that is the only genotype that will give you brown chicken. Then after you do that, you're asked to summarize again more concisely, or all the different genotypes that can result in a yellow chicken. And this explains the reasoning leading up to that. If you answer correctly, it will turn green. If you answer incorrectly, it will turn red. Here we've answered that correctly. And this is, and then after specifying the genotype, you describe what will happen if you have one of those genotypes. And So this is what the prediction lesson for gene interaction looks like, where you know what the underlying properties are, and you're determining what the phenotypes, what the data would look like given those. In the experimental version of gene interaction, you are given 3 different strains, in this case 3 different types of cats, brown, black, and gray, and you're asked to cross them to look at the offspring. And what you are given when you do that, you are given the data, and you're asked to conclude something about the genotypes of these different strains of cats. And then after you collect all the data, you can determine what the genotypes are for these 2 genes, and then you go back and fill in what the genotypes were for the crosses, which you did not know as you were doing them. So you elaborate all of this as a result of these 3 experiments you performed. And then after you do this, you can then guess as to how those 2 genes were interacting in the pathway. So in the second topic, gene regulation, students read a problem statement describing how a particular gene is regulated, how it's turned on or off. They summarize. The first thing they do is they summarize this system, and then they look at, again, the underlying parameters. In this case, they look at several genotypes, several different mutations. They first characterize those mutations, and then with a menu, they select what the impact of that mutation is on the system. And then after that, they determine whether the system would be functioning correctly. In this case, when a particular molecule that turns the system on or off, an external molecule called an effector, comes in, if the system is not functioning properly, then it would always be off. So this is the forward reasoning version of this lesson. The backward experimental version of the lesson, the student is given a system with 3 genes that they can manipulate, and they need, just as in gene interaction experimentation, they perform several crosses, they look at the data, and based on the data, they draw inferences about what kind of system, what kind of regulatory system this is, and what the roles of the specific genes are. In the developmental genetics lesson, which introduces signaling pathways, the student again is shown exactly what the underlying properties are and is asked to trace through the individual states of the signals within a signaling pathway. And in some cases, there are mutations that break the pathway, so that that the molecules in the pathway, the signals, are permanently off or permanently on based on that mutation. In the backward version, or the experimental version of this lesson, they're given a set of strains, and by performing crosses, by performing experiments, they learn about how the genes are ordered in a pathway, and whether each step in this pathway is activating or inhibiting the next step in the pathway. This different type of link means that, for example, that B inhibits the next signal. in the pathway that it turns it off. In the cancer pathways lesson, again, there are a number of mutations that are described in signaling pathways that are involved in the cell cycle to turn the cell, to stimulate cell proliferation, cell growth, or there are also pathways in the cell cycle that turn it off or kill the cell if it's broken, if there are mutations that make it unable to function. So this allows the student to look at a cell with a collection of mutations to determine ultimately whether there is a change, an increase or decrease in cell proliferation that might lead to cancer. After they've done the predictive or forward reasoning, then there's an abductive form of that lesson, an experimental form of that lesson, where the student is told that you have overactive, for example, overactive or underactive, in this case, overactive cell proliferation, and they're asked what change, what mutation in this particular signaling protein could have caused that. And ultimately, the student is asked if they were trying to design a cancer treatment for a patient who had a cancer with a mutation in that gene, how they would treat that particular, this particular mutation. Modern cancer treatment involves making a cocktail with a collection of drugs that treat different mutations in a particular patient's cancer. So in this case, this asks the student to think about the different ways that scientists design cancer treatments customized to a specific patient. Well, I hope that that this wasn't all too overwhelming. And again, all of this, we have shown that while this tutor was designed for college biology majors, primarily at Carnegie Mellon, but other places around the country, we have introduced this very successfully at several high schools, and high school students in AP biology and honors biology are very capable of learning this, and the learning gains that we have seen are comparable to the learning gains that we've seen in biology majors at several universities. So we are very optimistic that this, and we've just recently started introducing this to freshmen at Carnegie Mellon, including art and drama students who are not normally predisposed to learning logical representations, and they seem to be doing just fine with this. So we are optimistic that very large numbers of students will be able to learn the logic of pathway algebra and develop a deeper understanding of biology, biological reasoning, and what biologists do as a result of that. So I hope you've enjoyed this, and I hope it made some sense to you. Thank you so much.